Welcome to The Explainer. I am absolutely thrilled to dive into today's topic with you. Modern biology has this amazing but kind of daunting characteristic. It just generates massive, staggering amounts of data. Whether we're looking at gene expression, mapping out metabolic pathways, or tracking disease progression, we are literally flooded with millions of data points. But here's the monumental task, right? Finding the hidden, useful, and biologically relevant patterns within that absolute chaos. It's like finding a needle in a digital haystack. Today we're going to break down a brilliant study from BMC Bioinformatics that essentially rewrites the rulebook on how we find the signal in all that noise.
Okay, let's take a quick look at our roadmap. We'll start by looking at the messy world of data, identify a major blind spot in our current tools, introduce the upgraded LS NMF algorithm, actually put it to the test with real data, and then wrap up with what this means for the future of discovery.
So, starting us off, Section 1, the messy world of data, dealing with biological chaos.
To set the stage here, we really need to understand the specific type of information scientists are dealing with. Imagine you're a detective, okay? You walk into a massive room where 1,000 different conversations are happening all at once, and your job is to figure out exactly which people are working together on a specific project. How do you find a reliable pattern when the data you're listening to is just inherently chaotic? Well, that is the exact challenge geneticists face every single day. They aren't just looking at one gene at a time, they're looking at thousands.
A really prime example of this is microarray data. Basically, microarrays allow scientists to measure the expression levels of thousands of genes simultaneously across a bunch of different conditions, like varying temperatures or different chemical treatments. But here's the messy reality of biology. It's noisy. The data has incredibly high variance because some genes are just much more tightly controlled by the cell than others. However, and this is a crucial clue for our detective story, scientists can actually calculate individual estimates of uncertainty for almost every single one of those data points. We literally know exactly how noisy each specific measurement is.
That brings us to Section 2, the NMF blind spot, the flaw in our current pattern recognition.
For years, we've relied on a standard mathematical tool to try and solve this biological puzzle, but as you'll see, it has a rather fatal flaw. This standard tool is called non-negative matrix factorization, or NMF for short. Simply put, NMF is a machine learning algorithm. In the context of our messy microarray data, think of it as a grouping tool. It looks at this massive grid of genes and conditions, and it tries to find metagenes, basically clusters of genes that behave similarly across different conditions. The whole idea is that if genes are turning on and off at the exact same times, they're probably biologically linked and working together.
To pull this off, standard MMF creates a model and then checks how well that model actually fits the messy data using what's called the root mean square distance, or RMSD. Now, do me a favor, please don't panic at the math here. All you really need to know is that this equation is simply NMF's way of calculating the total difference between its neat little model and the actual chaotic biological data. It's just an error calculator, and obviously, the lower the error, the better the model.
But here is the absolute crucial point. Standard NMF trusts all data points equally. It completely ignores the measurement precision. Remember our detective analogy from earlier? Well, standard NMF is a detective that picks up a crystal clear, highly reliable fingerprint and then listens to a sketchy, anonymous rumor from a super unreliable source and treats them both as equally important clues. By relying on that basic RMSD equation, standard NMF entirely ignores those individual estimates of uncertainty that we know we have.
So what do we do about this? Section 3. Enter LS NMF, factoring in the uncertainty.
A really brilliant team of researchers, Wang, Consequently, and Ox, realized this flaw and decided to build a better detective. They created least squares non-negative matrix factorization, or LS NMF. Let's look at the stark contrast between the two. While standard NMF uses RMSD and blindly treats all data equally, LS NMF uses a chi-square error measurement. This is a game changer because it actually factors in the variance. It explicitly weighs the highly reliable, precise data much more heavily than the noisy high-variance data. It finally puts those uncertainty estimates to work.
And here is exactly how they did it. This is the modified equation. Notice the only real difference is that little sigma symbol sitting right there at the bottom of the fraction. That sigma represents the uncertainty measurement for each specific data point. By dividing the error by the uncertainty, you penalize the algorithm way more for getting the reliable data wrong, and you kind of forgive it for getting the highly uncertain data wrong. Think of it like a teacher grading a class. This new equation forces the algorithm to put massive weight on a heavily proctored, highly reliable final exam and put way less weight on a quick, chaotic pop quiz.
Moving to Section 4, putting it to the test, proving the power of LS NMF.
The theory sounds fantastic, right? But does it actually work in practice? The researchers applied both the old NMF and their new LS NMF to a massive real-world biological dataset known as the Rosetta Compendium. In this chart, we're looking at the overall error on the vertical axis and the dimensionality, meaning the number of patterns the algorithm is searching for, on the horizontal axis. That solid black line, that's our new LS NMF. Notice how the LS NMF error drops beautifully and consistently as the dimensionality increases. It's fitting the Rosetta Compendium data significantly better simply because it is actively optimizing for the known variance.
Now let's see how this translates into something honestly really clever. This chart reveals the surprising genius of LS NMF. Look closely at the dashed line labeled bottom 25% RMSD. That line represents the absolute worst, most imprecise data points in the entire set. Notice how that line is basically flat? The error for those sketchy clues doesn't improve at all as the algorithm gets more complex. Why? Because LS NMF is intentionally ignoring them. It looks at the high variance, says, yeah, I can't trust this, and focuses all its computational energy on fitting the reliable data instead. It's a feature, not a bug.
But here's the ultimate question. Does fitting the data better actually mean we learn more biology? Absolutely yes. Direct your attention to the solid black line in Chart C. This is an ROC analysis which tests how well the algorithms actually recover known, verified biological co-regulation groups. Basically, did it find the right genes working together in a metabolic pathway? That solid black LS NMF line sits consistently higher than the dashed and dotted lines of standard NMF and basic k-means clustering. In fact, LS NMF was 15% better at recovering these known metabolic pathway genes.
Now, what's really wild about this next slide is that it proves the incredible stability of this new method. Here, they tested if the algorithms could predict functional relationships between genes based on the MIPS database. The solid green lines are our new LS NMF, and the dotted red lines are the old standard NMF. Just look at the dotted red line at the very bottom. That represents standard NMF running at 15 dimensions. It practically failed to give meaningful results at all. But the solid green line for LS NMF at 15 dimensions, it maintained incredible robustness and vastly outperformed the standard method. Because LS NMF isn't getting confused by all the noisy data, it doesn't just crash when you give it a slightly less than perfect number of dimensions to look for.
Finally, Section 5, the future of discovery, beyond the microarray.
So we've looked at a bunch of charts. Let's zoom out and bring this highly technical data back to the big picture. Why does upgrading this one math equation matter so much to the scientific community? Well, it matters because of the massive advantages summarized here. First, LS NMF integrates uncertainty directly and elegantly into its updating rules. Second, as we just saw, it vastly improves our ability to recover actual biological knowledge, finding real functional links between genes that other algorithms completely miss. And third, and this is a point the researchers made very clear, it operates with a minimal extra computational cost. It has the same conversion speed as the old algorithm. Guys, we get all these amazing biological insights without needing to buy massive, expensive supercomputer upgrades. It's really just smarter math.
Which leaves us with this final, really provocative question. What other scientific fields could be revolutionized simply by telling algorithms what not to trust? The authors of this paper explicitly suggest that mass spectrometry could be the next frontier for LS NMF, since it also produces data with really clear variance estimates. By teaching our machines to weigh the quality of a clue rather than just the existence of one, we aren't just cleaning up microarray data. We are opening the door to faster, more accurate, and more profound scientific discoveries across the board. Thank you so much for joining me for this explainer. And remember, keep questioning the data.